Why is EPS essential for biofilm formation? Imagine tiny microbes building their own microscopic cities, complete with walls and defenses. That's what happens in biofilm formation, and a key player in this process is something called extracellular polymeric substances, or EPS. Think of EPS as the glue that holds these microbial communities together. It's a slimy, sticky mixture made up of sugars, proteins, fats, and even bits of DNA that microbes secrete into their environment. Without EPS, these tiny creatures would be like loose leaves blown away by the wind, unable to stick to surfaces or each other. EPS acts as a sturdy scaffold, giving microbes a foothold on surfaces like rocks, pipes, or even your teeth. It helps them attach firmly, transforming scattered cells into a structured, multi-layered community. This sticky matrix creates channels that allow nutrients and waste to flow in and out, keeping the community alive and growing. It's like a tiny highway system inside the biofilm, delivering supplies and removing trash. From a bizarre biology point of view, EPS turns simple bacteria into tiny fortresses. It shields them from threats like antibiotics, immune system attacks, and environmental stress. The slimy barrier can trap antimicrobial agents and immune molecules, making it much harder to kill the bacteria inside. That's why biofilms are often resistant to treatments that wipe out free-floating bacteria. EPS also encourages social behaviors among microbes. Because it holds cells close together, it promotes chemical communication called quorum sensing. This allows bacteria to coordinate activities like defending against threats or sharing resources. Sometimes, these communities act more like tiny multicellular organisms than individual bacteria. A strange but important part of EPS is extracellular DNA. Once thought to be just leftover genetic material, it actually helps keep the biofilm stable. Some bacteria releases eDNA on purpose, using special vesicles to do so. When scientists treat biofilms with enzymes that break down DNA, the biofilm often falls apart. This shows how vital eDNA is to maintaining the structure. In practical terms, EPS is a prime target for fighting biofilms. Enzymes that break down its components, like DNA or sugars, can weaken the biofilm's defenses. Once the slime is broken down, bacteria become more vulnerable to antibiotics and immune responses. It's like breaking down the walls of a fortress to expose the defenders inside. So, in the world of tiny microbes, EPS is the secret ingredient that makes biofilms so resilient and bizarre. It builds the physical and chemical environment that keeps these communities alive, protected, and capable of complex social interactions. Without EPS, biofilms would not exist as we know them, and these microscopic marvels would be much less formidable.